Okay, so we're going to pick up where we left off on the last video. Hopefully this loads pretty quickly here. And we're going to talk about chemical digestion. And most of what we're going to talk about with chemical digestion is how do exactly these digestive enzymes work. Now, an enzyme is very important to remember here. An enzyme is going to allow for a chemical reaction to occur much faster because they're catalyzing that chemical reaction. The idea behind that is that they're lowering what we call the activation energy, which means that the chemical reaction can occur much faster. So we look at them as proteins. Again, remember, if we talked a little bit about this, especially in microbiology, about proteins, okay? So what we're going to talk about here is they're specific. We're going to talk more about how they're specific in their actions, but in addition, they also only operate at specific pHs depending on what protein you're dealing with, okay? And the idea here is that they're breaking things down, so breaking our carbohydrates down, breaking our proteins down, breaking our lipids down, all of these things that we ingest in our food. They're breaking down in the form of hydrolysis because all of them have at least some type of oxygen, hydrogen. So therefore, we're going to be removing water molecules in order for us to get the good stuff out of each of those things. Okay? So again, they're going to catalyze a chemical reaction. They consider it in both directions, but for the most part, we're only going to be looking at one direction. Okay? So don't be super concerned about that. They're continuously being destroyed and synthesized. So if we don't have those, then we can't, obviously once that protein stops working correctly, we'll get rid of it, and then we'll make another new protein to be able to do that. And our cells do this through, transcription and translation, okay? And most digestive enzymes, we consider them to be proenzymes when they're inactive, and then once we need more of them, they become activated, and now we have more digestive enzymes, okay? So here's looking at the effect of pH on our digestive enzymes. Remember, back here we said function optimally at a specific pH, right? So each one of our digestive enzymes, depending on where they're located, are going to work at a different pH. Now, our pH of our stomach is probably around 2 to 3 because we have this thing called hydrochloric acid within our stomach. It's a very strong acid, and it brings our pH down. So if we look at pepsin here, this is going to have an optimal range here. Wherever our peak is going to be between around 2 to 3. And so that means that pepsin is going to work inside of our stomach. That's where you would find pepsin. Now, when we go to once we get down to our small intestine and large intestine, they're back to normal. They're around a 7 pH, around there. We can kind of go anywhere in the neutral range between 6 to 8 is about what we would see as considered a neutral pH. And that's what we're seeing here for tripepsin. They're going to be between 6 and 7. So that means that we're going to find tripepsin in our intestines. And again, mostly what we're concerned when we're talking about chemical digestion, we're going to be talking about our stomach and our small and large intestines, because that's where we do the most of the chemically, breaking down of our food. Okay. Up until that point, all we're doing is doing a little bit chewing, putting a little bit of saliva on there. They do have some digestive enzymes in there to help break it down, but we're not getting the stuff out of it yet. Okay. So up until this point, up until we hit the stomach, we're not getting anything out of our food. We're just breaking it down, getting it to the point where we can get something out of our food. Okay, so the first type we're going to look at is going to be carbohydrates when we're looking at digestion, okay? So carbohydrates are typically polysaccharides, and we can't really use those polysaccharides. Again, if you've been in microbiology, you would have also learned that sometimes even those polysaccharides that we eat, especially in lettuce and all of our vegetables and, any of our plant-based food, which are your vegetables and our fruit, a lot of times those ones can't even be broken down by us. We have to have additional, bacteria or, things in our normal flora, which are totally normal, totally safe for us, to break down those polysaccharides into disaccharides, okay? But for all of the carbohydrates that we can break down ourselves, we're going to look at this thing called amylase. Now, we're going to see a couple different types of amylase when we're looking at this digestion, okay? But there's the salivary that's going to be found in both of our saliva. So that's where we start to break down these polysaccharides. And then also we're going to be secreting it from our pancreas as well, okay? Because our pancreas is considered looking at our blood glucose levels, if we remember from chapter 26. Okay, and we're talking about all of our endocrine system. This is part of the reason why they control our blood glucose levels. Okay, they produce this amylase, pancreatic amylase, that breaks down polysaccharides to form disaccharides. Now, disaccharides are typically sucrase or sucrose, sucrose, lactose, and maltose. And then from there, they're going to be digested again by different enzymes in order to become glucose and fructose and all those things that we need. Okay, so sucrase, lactase, and maltase. Okay, the only difference between any of those is just what is involved. So sucrose is going to be two glucoses. Lactose is completely different. Maltose is different as well. Okay, so that's the only reason why you have to have different enzymes in order to be able to break them down, because you have different monosaccharides that bond together to form the disaccharides. Okay, so again, we're looking here. We're going to be making this, we're starting our salivatory, so start with our saliva, and then eventually when we get down into our intestines, we're going to finish it off with our pancreatic amylases. So again, these perform hydrolysis on the polysaccharides to break them down to make disaccharides. And the different disaccharides that we have may be lactose, sucrose, maltose, any of those would be considered disaccharides. Then from there, we're going to break them apart again through hydrolysis. And then we're going to make monosaccharides. Okay, so those are going to be our single sugars. Does this make sense? Hopefully it does. Okay. The next step we're going to deal with is going to be dealing with protein digestion. And again, we need proteins as well. We don't just make amino acids on our own. Okay, that's not something that we're really good at making. Sometimes we do, but for the most part, we get them from our food. That's why it's important that if you're lifting a lot or doing stuff that requires a lot of energy, you should have some protein in your diet. Okay, because those proteins are going to build up to make those enzymes that will allow for you to be able to do those chemical reactions quicker and therefore allow for you to have better stamina. Okay, so when we're looking at protein compounds, we want to get them down to our amino acids. Just like we wanted to break down our carbohydrates into those single monosaccharides, we're doing the same thing here. We have a long chain in these protein compounds of amino acids, so we want to break them down into our single amino acids that we can use to make new proteins within our cells. Okay? So typical group that does this is going to be called our proteases. Now again, remember, anytime we see this ASE, we're going to be dealing with an enzyme of some sort. Okay, so anytime you see ASE, it's an enzyme. Okay, so they're going to basically take those proteins, break them apart essentially. So going to break down the protein folding, get them down to a single structure, and then eventually get them down into our amino acids. Because remember, if we're a little bit of talking about proteins that we've done, we know that we've got our single first structure is going to be our primary structure. Then we've got our secondary structure, tertiary structure, and quaternary structure. Okay, so we're going to go through all those levels until we get down to our single amino acids. So the main ones that we deal with a lot when we're talking about this, obviously pepsin. That was the whole reason why we showed that to you at the beginning here. So that's found in your gastric juice within your stomach. Okay, next one's going to be tripepsin, which is going to be, or trypsin. Sorry, I spoke that wrong. Which is going to be found in the pancreatic juice, which is found in your small intestine. Okay, because whenever you secrete that pancreatic juice, it goes right into your small intestine, and that's where you'll break down stuff there. And the other one is peptidases. And again, these are made by your intestinal mucosa, intestinal brush border, so around that area, and you'll be finding them in your small and large intestines. Okay, so again, we're looking at this, we're going down. We just want to get these peptides down to the point where they are broken down into amino acids. Okay, that's the whole idea. We're getting these big, strong proteins. We're breaking them down till we get to our single amino acid. Okay. The last type that we're going to really talk about here is going to be fats. And that kind of makes sense because the only other thing that we really have that we get out of our food for the most part, we do have some nucleic acids. We're going to briefly discuss those. But for the most part, what we're going to get out of our food is going to be lipids. And again, this is where, remember, we talked a little bit about those where we have this, big thing called bile, and where it's stored. We've previously talked about that. Okay. So In order for any of our fats to be digested, they have to become essentially a bile salt, which means that they have to be encoded in bile, which occurs by being secreted from your, does anybody remember? It's from your liver, and then we're going to be stored essentially in your gallbladder, which is why your gallbladder isn't necessary, because you just are storing an extra bile. Okay, so it's kind of there. It's good if you use it, no big deal. Okay. So we're going to talk about, so basically what that does is then we can allow for this thing called a pancreatic lipases, or any of our lipases in general, to be able to recognize these fats, these lipids that have been made into bile salts, which is what they're called after they've been encoded in bile, which allows for them to recognize it, to break it down. Because fats in general, we don't recognize. Again, that's why, if you eat too much fat, if you have a high fat diet, you're going to gain weight, right? You're going to gain weight in the form of fat. Okay? It's because you only make so much bile to be able to digest appropriately. Okay? So that's going to be, the rest of it, you're just going to kind of be eliminating. Okay? You're going to be kind of keeping on there. Okay? So for the most part, we're going to deal with whenever we have any type of part of our food that doesn't really fall into any of these categories. Okay, we're going to talk about that. It's going to be kind of going through as feces. Okay, so what we kind of, and that mostly happens in your large intestine. And essentially what that does is it allows for us to go ahead and eliminate them and, essentially get rid of the residues that we don't need anymore. Okay, so again, this is just this idea of when you're looking at fats, again, big reason why they don't really work, so why it's a little bit more difficult for us to recognize them is because a lot of times they're oil-based or hydrophilic and hydrophobic. As you can see in this being shown here, okay? This is a micelle, which can be found within fats, okay? And the idea here is that you've broken down your fats, okay, no big deal, but you have a hydrophobic end or hydrophilic tail or hydrophobic tail and a hydrophilic head, which means this likes water, so it's going to basically form this circle here. And that's because of the fact that you have an area that doesn't like water and an area that loves water, okay? Okay, this is more fat digestion. I hope you guys understand that. There's big one that we're going to talk about here is pancreatic lipase, but any type of lipases will go ahead and break down your fats, just so everybody knows. Okay, so how do we go ahead and get this stuff out here? This is where we talk about secretion. And whenever we're talking about secretion, it's more or less to get the stuff out that we need to be able to break down our food. So the first thing we're going to secrete is always going to be our saliva, our spit, essentially. And that's because it contains that amylase that starts the whole idea of being able to break down our food, okay? So that's a small amount, and the only thing is it starts to break down those starches, makes them into smaller particles. Okay, we're not really gaining anything out of them. We're just breaking them down into smaller, essentially disaccharides. Okay. The next is going to be the gastric juice, and that's obviously given off by our gastric glands within our stomach. Okay. And so what happens here, we're going to be talking about is essentially the big thing that's going to be found in the gastric juice is the pepsin. And what does pepsin do? It breaks down proteins. Okay. The other major thing that we have within our gastric juice is hydrochloric acid. And that's what they consider secreted by peripheral cells. Okay. And essentially what they're doing here is we're going to basically decrease the pH of our stomach acid or gastric juice in order for this pepsin to work. Because remember, what pH does pepsin work at? Between like 2 to 3 is the most optimal pH. Okay. And again, whenever we're talking about acids, this isn't something that we talk about a whole lot in this class. We will talk about acid-base balance later on, which kind of gets into this a little bit. But the idea here is that anytime you have hydrochloric, any type of acid, you're going to be giving off a hydrogen ion. And essentially a hydrogen ion can also be called a proton because all it has is a proton in it. It doesn't have any neutrons. So that's why they call these proton pumps. And essentially that's where we regulate how much we're secreting of that hydrochloric acid in order to maintain the proper pH. Because if you add too much hydrochloric acid naturally has about, anywhere from 0.5 to 1 of a pH, which is much too low for a pepsin to work properly. Okay, so this is just showing that. Okay. In addition, castor juice, sometimes they also are basically giving off intrinsic factors that will protect vitamin B12 because that's something that you can also get out of your food. And again, this is something you're going to break down yourself. Your normal flora will do that for you. But again, you have something that protects it from that hydrochloric acid and anything else that could possibly break it down. Okay. And again, we're looking at only here, we're trying to make chyme. Okay. The next type of juice that we're going to be talking about secreting is going to be our pancreatic juice. And this is going to have all different types of goodies in there, specifically proteases, lipases, nucleases. So those are going to be anything that breaks down different types of enzymes or any types of food that may have some nucleic acids, because again, don't completely make 100% nucleic acids. Most of them we do have, but again, we might need additional ones, and therefore we're able to use those. Okay, specifically for RNA more so than DNA. You're not going to see that as much for DNA. Okay, and amylases, those are the ones that just go ahead and get rid of starches. Okay. Okay, so big thing we talk about here, again, this is all going to be based off of a chemical balance that we want to keep here. So we're going to make sure that we always, and then most of it has to do with bicarbonation. Okay. Okay, next thing, bile. So this is going to be considered, this is going to be secreted by your liver. Okay, so FYI, your gallbladder does not make bile. Your liver makes the bile. Your gallbladder stores it. Okay, and so therefore, these biles encase themselves in shells to form tiny spheres. And essentially what they do is they allow for us to recognize those fat cells that we can't recognize otherwise. Okay. So essentially what can happen here is that this is now going to be called micelles. So essentially that's what they're forming when we showed this earlier. Right? Okay. So essentially what they're doing is making these essential micelles, okay, because the oil base is the fat, essentially. Okay. And this allows for it to be broken down. Okay. So that's a big thing of bile. So what can kind of happen with that, we can have some cholesterol. It's going to be kind of like a byproduct that we form after the fact of breaking down our fats. We can also have some bile pigments. Basically, they're going to be excreted by the liver, and eventually we're going to eliminate them in our poop. Okay, intestinal juices. So this is going to be anything that's going to be given out by our intestinal mucosa. So a lot of that's going to be mucus and water. We're going to again get some more mixing of the chyme. That's basically how we break things down. Okay. And we can also release some sodium bicarbonate. Again, sodium bicarbonate is going to be a base, so that'll actually increase the pH. Because when the chyme normally comes into the intestines, it's going to be at a pH of about 2 to 3. And most of the enzymes that work in the intestines are going to be at a pH of about 7. So a release of sodium bicarbonate will bring that pH up closer to a pH of 7. Okay. So again, this salivatory secretion, this is something that a lot of times whenever you you think you're salivating at something, this is because you're more or less it's like a reflex. You're going to see essentially some type of mechanical or chemical stimuli in the presence of food. So when you see food or you smell food, those are going to be essentially going to allow for you to start getting out that saliva. You're almost salivating at the food because that's your indication that you're hungry. And that's all that we're talking about here is essentially you have some type of chemical or physical or mechanical stimuli. So you either see it or you smell it, and then you want to eat the food. So therefore you start salivating. Start giving off that saliva because you want to start breaking down those carbohydrates immediately as soon as they enter your mouth. When we talk about the gastric secretion, okay, again, whenever we're looking at this, we're going to have 3 different phases in which we can go ahead and essentially, I would say, back off a little bit on our gastric juice secretion. First one is going to be the cephalastic phase. This is the psychic phase, where basically the idea here is that you've got some type of stimulus, nerve stimulus, okay, that allows for this gastric secretion to start occurring. So usually it's an involuntary response, but it's controlled by by your brain. Okay, so you're not really noticing that you're doing it, but you're thinking, I just ate some food. Let's go ahead and start secreting some of those gastric juices. Okay, the gastric phase. So the idea here is whenever you have digestion that reaches the pyloric portion. If you remember Helicobacter pylori, they're going to be the ones that give you stomach ulcers. So this is a way that you can go ahead and make sure that that doesn't happen. Okay, and the intestinal phase. So this is going to be, you want to make sure that the idea here is that you're getting enough chyme to pass through and that the chyme is staying that milky, liquidy color. You want to make sure it stays a liquid, obviously. But that liquidy color. So that's where the intestinal phase there, that'll control as well. So the pancreatic secretion, this is going to be stimulated by hormones that we have secreted from our intestinal mucosa. And the big one that's going to be here is something called CCK. Oh, I lost it. Okay. CCK, which is going to be I can't say this, but you can look it up in your book. It's on page 918. I'm not going to try to say that one, unfortunately. I will probably butcher it. Okay. But the idea here is that this is going to, it's going to let you know that we need to secrete more of our pancreatic secretions because it'll notice the presence of fats, carbohydrates, proteins, all those things that you need to be able to get out of your get out of your intestines, okay, and out of your food, okay? Okay, and then we're looking at bile. Again, bile is continuously secreted by the liver. So that's why, again, technically you really don't need your gallbladder, but bile will constantly be secreted by your liver. And then it's just stored essentially in your gallbladder and released at appropriate times. We don't really know a whole lot about intestinal secretion. Again, part of it probably has to do with your normal flora, but there's no real evidence to suggest that. So again, not really sure. The idea here is that it's stimulated to release hormones that increase the production of intestinal juices. That's the only thing we really can even suggest when it comes to intestinal secretion. Okay, the last thing we're going to talk about is going to be absorption. Okay. So this is going to be essentially where we go from intestinal mucosa to the blood. Okay, because how do we get places, or lymph? How do we get places in our body? Either through our blood vessels or through our lymphatic system. Okay, so the idea here is that we're going to have some very complex mechanisms that go through this. And the idea here is that this probably is going to require some energy, which is where we get this active transport. Okay, so we have the first one would be secondary active transport. So that's going to be how our sodium is going to be transported. Essentially what will happen here is that we have a sodium potassium pump that will be put on our intestinal mucosa. And then from there, sodium will be kicked out of the cell and kicked out of the epithelial cell along the intestinal mucosas and go ahead and go to the blood capillary. That's essentially what it is. We want to talk about sodium co-transport. This is going to be dealing with how glucose is transported. And the idea here is that we're just going to be looking at having the sodium be attached to the glucose. The sodium allows for it to go through the sodium potassium pump, and out it goes, and then it gets absorbed by your blood capillary. Okay? Now, there's other ways that mostly what we talked about there was dealing with mostly monosaccharides, right? Well, we also have other ways that we can get this through transcellular absorption, which moves nutrient particles through the cells. And again, most of this is going to, again, involve active transport of some sort, okay? So that's the idea here. All of this is going to travel via active transport to your blood cells. And then from there, or your lymph. And from there, it'll go to the rest of your body. Now, the big thing that we need to talk about with monosaccharides and amino acids, they're going to go to your liver first, and then go ahead and go to the rest of your body through your liver secretions. Okay? When we talk about elimination, basically what the idea here is that you've got everything's going to kind of be built up into what we call feces within your large intestines. Okay? And that's going to be from the digestive tract. And this is going to essentially, it's going to go through a reflex called defecation. And no, this is like, I mean, most of the time this has a bad connotation next to it. But what we're going to talk about here is literally you already know it's going to happen. You're on the toilet and we're all good to go. Okay, but the idea here is that we're going to have some stimulated receptors because we know that we need to go to the bathroom in the rectal mucosa. And then this essentially what it's going to do is cause for distension of the rectum, which allows for you to essentially push out the feces that you have that you need to eliminate from your system or expunge from your system. So there's two different things that can happen with this that can be kind of problems. The first one is constipation. And essentially what happens is that the colon and the rectum move at a slower than normal rate, and therefore you can't poop as easily. And so you feel constipated. On the other hand, if you have diarrhea, you're going to have a little bit more of water and electrolytes within your stool, and therefore it's going to move a little bit quicker through there. And again, you're going to have, this is because of the increased motility of the small intestines. You're not absorbing as much as you should be. Any questions? Please let me know. Okay? So we'll move on to the next video then after this.